Okay, hi. So we are going to revise B1, and then I'll do a separate video for C1.1, which for my class we are testing together. So this is going to be carbohydrates, lipids, and proteins. So B1.1 is carbs and lipids, and then B1.2 is proteins. So we are going to do two sheets, but I am going to include some of the protein stuff on the first page, just because you have to know some of the same information, and because there's probably a little bit more on proteins than there is carbs and lipids, so we'll put some of it there. So we're actually going to do this first page as a giant chart, so I'll probably have to erase kind of halfway, but you can kind of make it as one chart. So the first half of the chart is going to be basically elements, monomer, polymer, and condensation, and then almost the whole bottom half of the page will just be looking at the specific polymers that you need to know, because you have a few kind of example ones. So that's just a way to set it up to try to organize it a little bit differently. So we'll make the chart. I'll probably have to erase a time or two, but you can just kind of keep going with your giant sheet. In theory, we could add nucleic acids. It is a biomolecule, and you have to know similar things. But since we already covered that in A1, I'm not going to put it on here as well, because it's already revised in that video, and it wouldn't be on like a test just on B1. But remember, if you're revising for your exams, that nucleic acids are the fourth biomolecule. So it would be a good idea to go through and say, do I know the elements? Do I know the monomer for it? Do I know how condensation reaction does it? Do I know the actual polymers? So you could kind of do a little revision check on how you're at with that as well. So we're just going to do the main three on this one. So we're going to set up a table. We're going to put biomolecule down this first column. I'm going to use most of my board just so I have enough room to not write tiny. And again, I use the page this way still, I didn't turn it. Then for each one, we need to make sure, and this should be pretty easy, that we know the elements that it contains. Then we need to talk a little bit about the monomer that makes it up, remembering that monomer means one part, so that's the subunit. And included in that, we're going to talk a little bit about the properties. So what are the properties of the monomer itself? And we'll include that. Then, like I said, we will do condensation reactions and polymers, but I'm going to wait and not draw the line yet because I want to make sure I know how much space to use. So you may just want to set it up like this for right now. Biomolecule, and then we are going to have our three biomolecules. So we're going to have carbohydrates, looks about a third. Then we're going to have lipids. Just trying to figure out how much board I need. And then we'll have proteins. Okay, so we're just kind of covering those three. So first thing for elements is we want to work out what actual elements are in each of the type. So for carbohydrates, they have carbon, hydrogen, and oxygen. And they have a specific ratio. So they should have around a 1:2:1 ratio. The key really is that for every carbon, you have twice as much hydrogen, but you do have an oxygen. So they have about a balance of carbon and oxygen. That's what's going to make them distinct from lipids. For lipids, they are largely carbon and hydrogen. So they are going to be long carbon-hydrogen chains. And then they're going to have a small amount of oxygen. And as we kind of draw that, we'll see what that looks like. But basically, there's a tiny bit of oxygen connecting the fatty acid to the glycerol, but then it's just CH chains. So that makes it distinct from carbohydrates, which have more oxygen. And then proteins are going to be carbon, hydrogen, oxygen, and also nitrogen. So they actually have an amine group that has the nitrogen in it. I want to put two little notes while we're kind of at this point. So we'll notice that in all of them, they all contain carbon. So we're just going to make a little note of that carbon is the kind of basis of all biomolecules. So it is kind of the building block of life for many reasons, but largely that it creates a lot of diversity because it can form 4 different covalent bonds, or it can form a combination of single and double covalent bonds. So it has a lot of flexibility in terms of its bonding powers that makes it a really good kind of building block or building element. So we'll just say can form 4 covalent bonds. And it can also have a combination of double and single bonds. So just a really flexible molecule that makes up kind of the basis of living things. The other thing I want to put a note of, like I said, I'm not going to go all the way into nucleic acids, but we will while we're doing this little thing. I'm just going to make a note that nucleic acids are going to be carbon, hydrogen, oxygen, nitrogen, and phosphorus. So they have a whole other element in them, and they're the only ones that always have phosphorus. So just a little reminder while we're on there. Okay, so that one's pretty straightforward. Now we need to look a little bit at what are the monomers, and we are going to do a little drawing of the monomer so that we are familiar with its kind of chemical structure and any important properties. So the monomer for carbohydrates is monosaccharides, which literally means one sugar. So mono is always one, saccharides is for sugar. And then of the monosaccharides, the one that you need to kind of specifically know about in detail is glucose. And you would need to be able to essentially draw glucose and account for its properties. I don't know that it doesn't specifically say you have to draw it in terms of knowing exactly where each H and OH is. So you would need to know it's like a hexose ring, or it forms a ring in aqueous solutions anyway. So that's generally how it's depicted. I'm going to go ahead and draw it in detail and have you kind of do that. Just in case you have to ever draw it, it's probably worth knowing that. So remember, it is going to be a hexose sugar. So it is going to have this chain here that actually has 5 carbons. We number it after the oxygen, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and then the 6th carbon actually comes up from carbon 5. So it's kind of a branched one. And then each of these have an H and an OH group coming off of them. So HOH. Then they're going to be HOH. This is actually going to be OHH, and this is going to be HOH, like that. So if you can kind of remember the general idea of that, a couple things to point out on this. If we were the way I've drawn it, I've actually drawn it as alpha glucose. If we were to make it beta glucose, we would flip that group there. So that is what makes it beta glucose, is the hydroxide becomes above instead of below. The other thing we want to make a note of, because you do need to know the properties, is that these OH groups are what make glucose polar. So those OH groups are polar, so they actually give glucose its polarity. And the polarity of glucose is quite important because that gives it some of its properties. So the fact that glucose is polar means that it is soluble and therefore transportable in water. So it's easy to move glucose around a living organism because it will dissolve in the water and then be transported that way. The other thing that's important is with all of those covalent bonds, it's a highly stable molecule, so it's really good that it's not reactive, but it has a lot of strong covalent bonds that have the ability to release energy. So it's stable, but it releases energy when oxidized. I'm just putting oxidized. We haven't really gone into what that means yet because we haven't done topic C, all of that. So anyway, when you're revising for your exam, that will make more sense. Right now, I would say if you're just learning this now, just focus on the energy part. So has a lot of energy that can be released, which is really important. So we have drawn glucose. It is a hexose ring. So hexose just meaning 6 carbons. And remember, it forms a ring in aqueous solution. So I'm just being lazy and writing in water. And because we are talking about living organisms and your body is an aqueous environment, that means it's pretty much in that form inside our bodies. There are also some pentose sugars, and examples of those include fructose, and also includes both deoxyribose and ribose, so that we talked about when we did nucleic acids. So those are sugars that are used in that example. Okay, so that's kind of everything you need to know about monosaccharide and specifically glucose. When we go on to lipids, they actually have two different monomers. So they have a glycerol, which is just going to essentially provide the backbone. So it is part of what builds a lipid, but we're not going to worry about that as much because the more significant one is that it is made up of fatty acids. So a fatty acid is a long carbon hydrogen chain, and there are two different types of fatty acids. So we're going to draw an example of each. So a fatty acid can be saturated. So if it is a saturated fatty acid, they are both long carbon hydrogen chains, and in this case, all of the carbons have only a single double bond. So you'll notice they all form kind of the four, one on the carbon on each side, and then they have the hydrogens above and below them. And so that is what makes that unique. That connecting one does actually have a double bonded oxygen and a hydroxyl group at the end, but then the whole rest of the chain is like that. So the key here is for the saturated ones, there are only single bonds between the carbons, like that. As a result, they pack densely, so they become more densely packed. So they kind of can pull together. Therefore, we use them more as body fat. So our body fat tends to have a lot of saturated fats. So we form triglycerides. We tend to use lots of that. Not always, but they tend to be used more for that reason. Because they are so densely packed, they are going to have a low melting point. And then because they have a low melting point, they generally are solid at room temp. So when we think saturated fats, you can think like butter. So if you leave it, it'll soften, of course, but if you leave it at room temperature, it will stay in kind of a solid form. The other type of fatty acids, and I'm just going to draw the monounsaturated, but you could draw polyunsaturated by just adding more double bonds. So if we say monounsaturated, it just means at least one double bond, and poly would mean more than one. So this time, it might look the same here, 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 but then it'll have one place where it will have a double bond, and those generally will cause a kink. So it will kind of alter its shape, and that's significant because that's why the properties are different, is actually a result of that, like so. And then these form one less hydrogen because they have the double bond. So it looks something like that. So again, if it's monounsaturated, that is one double bond. If it is poly, it just means more than one. So I would just have to draw that with multiple. Okay, so that is my monounsaturated fatty acid. Because of the fact that it has that kink, it causes it to have a kinked shape. Therefore, they are not as dense. So they're a little bit harder to pack in. We use unsaturated fatty acids quite a lot in the cell membrane. So a lot of times they might get used instead of being used to make adipose tissue. We might use them instead to regenerate cell membranes and healing and things like that. So generally they're thought of as healthier in our diet because we use them for a more diverse range of things. As a result, they are going to have a higher melting point. and they are therefore usually liquid at room temperature. And so our example of that would be like olive oil. So generally speaking, if you are eating animal fats, you are getting saturated fats. If you're eating plant-based fats, you're getting unsaturated fatty acids. Okay, so those are the two kind of categories for that. For proteins, we're just going to draw one general amino acid. So the monomer is amino acids. And remember that there are 20 different amino acids, and that of those 29 are essential, which means that we cannot make them at all, so we must eat them. The other 11 we could actually make as long as we have the elements. We could restructure them, but if 9's a really large amount, so basically you need to eat your protein. Okay, so then if we are drawing that, we have a central carbon. From that, we have our amine group. Then we'll have a hydrogen, and then the R group, which we'll talk about in a second, is what makes that amino acid unique. And then we have over here our carboxyl group, which looks like that. So you do need to know the names of these groups. So that is my amine group, and that is my carboxyl group. And then my R group is sometimes called the side chain, which is kind of a strange way of putting it. But remember, this is 3D, so it could move around, and that could be on the top, or the whole thing could flip, and that could be facing to the side, and these are like that. So just bear in mind they're 3 dimensions, so you don't have to always see the R coming off of the bottom. And then so like I said, this is what gives it its unique properties. So this is different for each of the amino acids. And it's also, because of its difference, it is what gives it its chemical properties. So it is kind of what determines that amino acid's chemical properties. And generally for our purposes, we're going to look at that as determines whether it's polar, non-polar, or charged. So those are kind of the 3 categories. You do not have to know any specific amino acids. So like here, I did draw glucose as a specific example. You don't have to memorize any specific amino acids. You just need to know that R group is what determines that. Okay, just so I don't have to erase as many times, I am going to try to squeeze in the last bit. I'm going to have to squat down a little bit, but at least I'm going to have to erase the chart once. Whoa. Okay, I'm just going to... like this. Sorry, you already wrote it. I'm just being... So this can be either polar, non-polar, or charged. Perfect. Okay, so last thing, and then we'll erase and do the bottom half of the chart. And I guess on mine, this is taking up a little bit more than half, so don't feel like you have to super cram it in. But kind of want to look at the actual condensation reaction itself. So remember, the condensation reaction is essentially the process that turns the monomers into polymers. So that is what we mean by a condensation reaction. And then at the end, we'll quickly cover the fact that hydrolysis is just the opposite of that. So if you understand condensation reactions really well, then you shouldn't really need to know much about hydrolysis other than flip it and reverse it all. And again, we already drew that in detail, so I'm going to try to keep this one a little bit more simple. So I'm not going to draw all the elements, I'm just going to draw the ends. So just my H and my OH. And then I'm going to draw my next one. So if I want to do a condensation reaction, I have to remove water. So as these are right now, they are perfectly stable, so they do not naturally want to react. So if you put 2 glucoses together, they don't just fuse. So they are actually perfectly stable as they are. So in order to destabilize them, I'm going to actually have to remove elements. So I actually removed the H off of the carbon 4 and the OH off of the carbon 1. I don't think you need to know that detail. You do need to know OH and H and call it a 1,4 linkage, but you don't necessarily have to remember which one pulls the OH and which one the H. But you might as well draw it accurately, like that. And then that is going to form what we call a 1,4 glycosidic linkage, because we pulled from the carbon 1 and then off of, remember, 1, 2, 3, so 1, 2, 3, 4, so we're pulling it off of this carbon 4, so that's where that came from. So we formed the 1,4 glycosidic linkage, and then remember, in doing so, we actually remove the water. So by removing an H and an OH, that creates a safe waste product. We will make a note, because it's going to come up on our next one, that we can also form 1,6 linkages. We are going to actually use those in a lot of the forms. So glycogen and amylopectin use an additional form. So that is also possible. But the main one that we tend to think of when we build the change is the 1,4 glycosidic linkage. Okay, then if we are going to build triglycerides, so I'm going to do an example as a triglyceride, and then we will talk about phospholipids as a second example. But generally, in order to build that, we are going to need a, like I said, a glycerol. So a glycerol is just a short little carbon hydrogen chain, like so. It is actually itself has the 1 to 2 to 1 ratio. So if it were on its own, it would actually not be acting as a lipid, but that's neither here nor there. The important thing is that we're going to add it. And then I'm going to draw these. I'm going to write it this way. You'll see why in a second. And then this is the long carbon hydrogen chain, and I'm just going to draw it like that. So sometimes you'll see it denoted that way. That's just basically a lazy man's way of not drawing all of those out. So long carbon hydrogen chain, and long carbon hydrogen chain. So dot, dot, dot. So if I'm doing a condensation reaction here, I'm going to pull the OH off my glycerol, and then I'm going to pull just the H off of each fatty acid, OH off the glycerol, just the H off the fatty acid, OH, just the H, and each of those is called an ester linkage. And then in order to make that one triglyceride, I did actually remove 3 waters. So because I did actually use 3, this is forming a triglyceride. If we wanted to, instead of doing that, if instead we wanted to actually form a phospholipid, we could, instead of using this, we can instead add a phosphate group. And that would give us a phospholipid. We'll talk about those below, but just being clear that they're actually formed the same way. But instead of doing 3 fatty acids, we do 2 fatty acids and a phosphate, but it's still formed with 3 condensation reactions. So exact same concept. And then if we come over here to our protein, we're going to do 2 amino acids. So again, I'm going to draw them like here. Great practice to draw them. You could actually get asked to draw an amino acid. And then we'll draw our next one. H, N, H, H. Perfect. And so if we are doing this one's pretty easy to remember, because if I look at it, there is only a hydroxide here. So I'm going to pull the hydroxide, I'm going to pull the H, and that should enable a bond to form between the C and the N. So make sure that you bond there, not between like the O's and so forth. So the bond should actually be between a C and an N. This bond here is going to look like that. So it is going to be an oxygen-based bond. And then same here, these bonds are going to basically be like COC, chain, chain, chain. So this one is going to form a CN like that. with it'll have that's here and here. So that is what the actual peptide bond looks like. Sorry if that's more confusing than helpful. I know when we practice drawing them in class, also because it's not visible, so that's link, there we go. So we draw them in class, we did have a bit of issues making sure we drew the C and the N connected. So that is forming a peptide bond. And then in doing so, we removed water. So hopefully by drawing that, we kind of clarified the relationship with water. So why do we remove water? It's in order to get the H and the OH, in order to make them reactive. Last little thing is just to quickly cover the opposite. So we'll just put over here, the opposite of a condensation reaction is hydrolysis. And hydrolysis is going to actually break a polymer into monomers. So if we are doing hydrolysis, we are taking a polymer and we are converting it into monomers. So that would be like digestion. So digestion is an example of hydrolysis where we're breaking bonds. And this time when you break them, if we don't return their H and OH to them, so to speak, they're going to stay reactive and just join back together. So we break them apart and then we need to give them back some elements to keep them stable. So when we do a hydrolysis reaction, we do add water. And we add the water so that then the H and the OH can kind of help stabilize the monomers. Okay, so that's kind of half our chart. Bottom half, we're just going to look at the polymers that were specifically listed that you need to know. So there's a handful of polymers that are kind of covered in the syllabus as polymers you need to know. So you just keep going with your chart. I'm just going to quickly erase. Okay. And there are going to be other examples of this, but what I'm working off of is the named ones in the syllabus. So these are the ones that you could be kind of specifically asked about, but particularly proteins, we're only going to look at 3 and there are way more examples of that. But in this kind of category, these are the ones that you were told to know. So I'm going to write over here, polymers. Okay, so for carbohydrates, there's like 4, and then there's like a division within that as well. So the first one, which is the one that has my division, that you have to know is starch. And when we talk about starch, there are actually 2 forms that it exists in. It does not actually say in your syllabus that you have to know amylose versus amylopectin. So just FYI, it used to be in the course, but I don't think they'll point blank ask you that difference. But given they have different structures, I am going to go ahead and clarify just in case you need to know it. So if we look at amylose, that is going to be only, so they're both going to be alpha glucose only. So alpha glucose, that's my alpha symbol. For amylose, they are going to have only 1,4 linkages. As a result, it's going to form like a straight linear chain, but then it twists into a helix. So only the one type of bond, but then it's going to twist into a helix. For this, also 1,4, but it's going to have 1,4 linkages plus some 1,6 linkages. So it's essentially using the 1,4 linkages as the main, and then those create kind of side branches. So it's going to have a branched shape. And they work together to make starch. So amylose plus amylopectin makes up starch. I think it's like 2080, might be 3070. You don't need to know the exact number, but the majority of starch is actually amylopectin, but some of it is amylose. So they work together to make up starch. And then the function of starch is to store energy for plants. And it can even act as a long-term storage for plants. And then of course, when we eat plants, we actually get energy that way. So a lot of times we eat plants for their starch, and then that gives us our kind of carbohydrate source when we break it down. Okay, so that is starch. The next one that you need to know about is glycogen. Glycogen looks very similar to amylopectin. So it's going to be structurally almost the same. So it just has more branches. So it is also, I'm going to write it out just so that we don't get confused by my symbol. It is also alpha glucose. It also has 1,4 linkages plus quite a lot of 1,6 linkages. So it's a little bit more branched than amylopectin. If you had to look at the difference between them, that's what it would be. So it has a very branched structure. And we use it similarly. So in our body, if we need to store energy long term, we do make fat. But for short term storage, we produce glycogen and store it in the liver. It's maybe a 24-hour supply. So basically the food we don't eat today that we might use tomorrow, we do store as glycogen. If we have more than that, then we use body fat because we don't have that much glycogen storage kind of area. So this is going to be short term energy storage in animals. And that's usually found in the liver. And so we'll talk more about glycogen when we do a little bit more on hormones and regulation, because insulin is in charge of converting between glucose and glycogen. So we can chat about it more at another point in time. And then we have cellulose. What makes cellulose unique is, first of all, it uses beta glucose instead. And it uses them kind of in a flipped pattern. So kind of in a flipping orientation. So one of them is right side up and the other is upside down. It's a very strange thing because if you flip beta glucose upside down, it's going to look like alpha glucose, but it is actually beta glucose flipped. So it's a beta glucose chain with a flipped orientation. So like every other one is facing a different direction. And that's going to be important for how it kind of works. It actually only has 1,4 glycosidic linkages, but then it also, so that's going to form a straight fiber. And then hydrogen bonds between fibers. And basically the hydrogen bonds form because of the flipping. They actually form between like where the 6th carbon is, because it's going to be up in one and down in the other, up in one and down in the other. So that's why it is a full flipping. So it's the OH and H flip, but so does where the 6th carbon chain comes. So it's going to have like carbon chain up, carbon chain down, carbon chain up, carbon chain down. And so hydrogen bonds form where the carbon chains are facing down. So that's probably more detail than you need to know. So you just need to know that the 1,4 linkages, and then it's not the same. They're not actually 1,6 linkages. They're hydrogen bonds formed between the fibers. So it ends up looking kind of like this, with like hydrogen bonds kind of connecting throughout. So long chains and then some hydrogen bonds attached chains, and that's going to make it a really like strong, durable material. So beta glucose, every other one has the upright orientation. So I might draw that real quickly because I'm worried I've made it more confusing. So remember that this has the CH2OH on it, and so then the next one is going to be flipped upside down. So that is now facing this direction. So it's going to have, that's what we mean by one of them. So then that is actually going to be facing down some of the time, and that contributes to hydrogen bonds forming. So have a look at a picture of it, because I'm going to be beyond my artistic abilities to draw the world's most beautiful picture of cellulose. But that's the general idea of it. And it is going to be used in plant cell walls. So its purpose is to form the cell walls. So it is a structural carbohydrate, and it makes up the cell walls of plants. Okay, and then last one that you need to know about that was mentioned was glycoproteins. And glycoproteins are basically a carbohydrate chain attached to a protein. And they're generally attached to a cell membrane protein. And we're going to do more on these when we get to the actual cell membrane. So this is just kind of a introduction that this is another form of carbohydrate, and their role is going to be in kind of cell identification. So an example of that is the ABO blood type. So what makes you type O versus type A type versus type B is which glycoprotein you have. So it identifies the type of cell. So that's an example of one. Okay, 3 different lipids that you need to know about, which we've kind of briefly mentioned. So you need to know about triglycerides. So triglycerides is going to be 1 glycerol and 3 fatty acids. So that's going to form like an E shape. A lot of the time they form what's called adipose tissue, or adipocytes, which are fat cells. So adipocytes are the cells, and then the cells make up adipose tissue, which is body fat. So that is quite simply body fat. So the reason why this works really well is because of the fact that they are non-polar. They actually do not dissolve in water. So non-polar, hydrophobic, which makes them amazing for storage. So if they were actually polar, they would tend to dissolve in cells. They would have issues related to osmosis. They would then pull a bunch of water in, dissolve in it, move elsewhere. But their non-polar nature makes them really ideal at staying put, which is excellent. They contain lots of energy. So you actually get more calories per gram out of fat than you do carbohydrate. So it's a really efficient storage because you can store more energy. And then they are also poor heat conductors. So that means that they trap heat. So they trap it, so they provide insulation. So body fat is energy storage, but also provides insulation for us in forms like blubber and things like that. Okay, the next example you need to know about is phospholipids. So phospholipids now are going to be 1 glycerol plus 2 fatty acids plus 1 phosphate group. And so what generally happens is the phosphate group kind of flips over. So imagine that's the glycerol and it ends up looking like that. So a phosphate group kind of on top kind of moves, reorients, and they end up looking like that. As a result, they make up the cell membrane. So they form plasma membranes. One reason they are able to do that really effectively is because they are amphipathic. And when we say amphipathic, we mean both nonpolar and polar in the same molecule. So the head is polar, the tails are nonpolar. So what they end up doing is they have heads facing both the outside of the cell and the inside, and then the tails have a region in the middle that is nonpolar. So that is how they form cell membranes. Cell membranes come up a few places in this unit, but we're going to kind of just mention it pretty briefly here because we're going to talk a lot more about them in another unit. So that's kind of what you need to know for now. And then the last category that came up was steroids. So steroids are quite unique. So they are basically 4 rings. So they become 4 fused carbon-hydrogen rings. So they're quite different from all these others. You don't really need to know about kind of how they're made. That's a little bit different, just that they are a type of lipid and they have a couple of important kind of components. So one thing is that they make up cholesterol. And cholesterol does lots of things for the body that are good, including play a role in the cell membrane, which we'll get to when we get to the cell membrane. But also they act as hormones. And they're quite effective as a subset of hormones because they're small and non-polar because of the carbon-hydrogen chains. And so they actually can enter cells. So they're unique from other hormones. Other hormones tend to be quite a bit bigger and have some polarity that make them hard to enter cells. They need external receptors, whereas our steroids are kind of a unique subset that have properties that can allow them to go straight into cells, which is good. Okay. Protein. So 3 examples of these. These again are just the 3 that are listed that you need to know about. I'm just going to move this slightly. Okay, so the 3 that you need to know about, first up we have collagen. And each of these represents a category. So collagen is an example of a fibrous protein, which means I can't spell fibrous, it does not have that first U. So that means they are like long and straight. They don't fold into like a spherical shape. So it looks kind of something, it has basically 3 polypeptide chains with like bonds between them. So it has 3 polypeptide chains. There's more detail, but in terms of what you need to know, you basically just need to know it as an example of fibrous. So I wouldn't worry about the detail of like repeating amino acid sequences and things like that. In terms of the function, it forms these strong and elastic fibers in the body. And so it acts as things like tendons and ligaments. So a really important kind of structural molecule. There's collagen in our skin and hair, things like that. So important kind of structural component. Okay, next one that you need to know about is insulin. And I'm going slightly out of order because we are going to go into, on the next part, we'll look at the actual protein structure. So that will come into this because this does have quaternary structure to it. Well, all 3 of them actually do. So this is going to be an example of a globular protein, which means it's going to have more of a spherical shape. And it is going to be non-conjugated, and that just means that it does not have any non-protein elements. So it's all protein. So it is just 2 polypeptide chains. And they're attached by numerous kind of bonds, including some disulfide bridges. And again, we'll talk about those in a second. So we're just going to write that for now, but we're not going to worry too much about what that is. We'll go revise that in just a second. And then what does insulin do? It is a hormone that decreases blood sugar by increasing the uptake of glucose. By the end of this course, you will know plenty more about insulin. So we're just doing a really brief kind of thing now for it. And similar to this next one that we're talking about, so much more to come. So just kind of the brief intro to it now. So hemoglobin, and I guess I did kind of draw collagen. If you were to draw this, it kind of looks more like with a couple of attached bridges. Hemoglobin is a little bit more complex, so it is globular, so it's going to have a nice kind of roundish shape, and it is also conjugated. And what that means is it has a non-protein element, so it has 4 polypeptides, which is going to be 2 alpha globin, 2 beta globin, and then it also has 4 heme groups, which are made up of iron. So they're not protein based, so they're made by something different. So it looks like 1, 2, 3, 4, 1, 2, 3, 4, with a big heme group in the middle of each one. Not the world's best drawings, but get the idea across. Okay, and then what does it do? So oxygen binds to it, so it is going to transport oxygen on the surface of red blood cells. Okay, so that's all of B1.1 plus a little chunk of B1.2 because we already did like what is an amino acid and we did those. So I'll erase this and then we'll do B1.2 and we'll do proteins, but our emphasis will be on the structure. So we've already included some of the background, so we'll kind of consolidate that part. Okay, since we already kind of covered a bit about amino acids as monomers and what an R group is and so forth, we're going to kind of go straight into levels of protein structure and then talk a bit about denaturation, and that will wrap up what we need to know about proteins. So we're going to make another chart, a little bit less dense, but just as another way of representing it. So this is going to be looking at levels of protein structure. And they do go in order. In other words, the primary structure is made first, then that is what then enables it to form in the secondary structure, then tertiary, and so forth. So we're gonna go across, so we're gonna write the level, then we're gonna write a description of it, then we're gonna draw a picture of it. And I'm not going to draw multicolored ones, so I'm just going to use purple for all amino acids just to make this a little bit easier. But feel free to use different colors if you want. And then we're going to do type of bonds. So looking at what kind of bonds hold together that example of that kind of structure. And then we're going to finish with impacted by. So what things could change that kind of protein structure? So that will be our chart that we are going to make. Okay, so, and again, I'll draw as lines as we go. So, okay, so let's start with primary. So when we say the primary structure, that is the specific sequence of amino acids joined together by strong covalent peptide bonds. So it is not just the bonds, but the sequence is really the key. So it is the specific sequence of amino acids. And remember, this sequence itself is determined by the DNA genetic code. And then it is held together by strong covalent bonds. And we've already talked about making those bonds as an example of a condensation reaction. We're also going to, in a minute, talk about anabolic and catabolic reactions, well, in the enzyme video that we're going to do next. And it's also an example of an anabolic reaction. So there's a couple different places this process has come up, but it is condensation that is actually going to do that. Okay, so when we draw that, again, I'm just going to draw generic, and we'll use a lighter color so that they're a bit more visible, dots. So when we are looking at primary structure, it's just a straight chain of amino acids, like so, held together by the strong peptide bonds. So that is our polypeptide, and we call it at this point a polypeptide. So we don't technically call it a protein until it's finished all of its forming and shaping and things like that. Okay, what are the bonds? So we've kind of already answered this, but the bonds holding this together are going to be strong covalent peptide bonds. And the key here, this is going to come up to what is it impacted by? because of the fact that these bonds are so strong, they are not going to be readily broken. So they're not going to be impacted severely by change in temperature and pH and things like that. However, because of the fact that this is determined by DNA, the primary structure of a protein is impacted by mutation. So if you change the DNA, you are going to indirectly, or not indirectly, directly change the amino acid. And we'll talk a lot more about mutation to come, but just while we're on proteins, it's worth going ahead and be thinking about that idea that mutations have an impact because they change the primary structure of a protein, which will have follow-on effects, which we'll get to in just a minute. Okay, so there's our primary structure. Next, we have a secondary structure. So what happens in the secondary structure is that we are going to form hydrogen bonds consistently between the carboxyl and amine groups that have become charged after the formation of peptide bonds. So when that bonding happens and they lose their H and OH, they develop a slight charge, and that slight charge is going to allow for kind of consistent bondings. So it's the consistent bonds So consistent H bonds between the actual slight charge left of the COO and the NH of amino acids. And again, the reason why they form hydrogen bonds is because this develops a slight negative and this develops a slight positive. So their slight charge that occurs from when they are forming the bonding is resulting in them able to bond. And it does so because it's consistent, it is forming a pattern. And a lot of times the secondary structure is less visible once you have a tertiary structure, but we've looked at the example of hemoglobin where you can still see the little helices. So there's two different types of patterns that can form. So in one example, they twist into a helix. So it goes like woo, woo, like so. The actual hydrogen bonds are here. And so that holds it into a helix. So that's going to be called an alpha helix. The other option is that they kind of turn into sheets. So in this case, it goes like, I find these strangely harder to draw. And they tend to pleat because of the pull of the bond. So it looks something like that. And again, this is still one polypeptide. So just to draw that, they might, let's make sure we connect them like that. So it zigzags and then loops around, and then the hydrogen bonds are here. So there's your hydrogen bonds. So this is called a beta pleated sheet. So that's my other type of secondary structure that can form. We already kind of answered what are the bonds. So we said here that these are going to be weak hydrogen bonds. These are not heavily impacted by mutation, so a significant mutation could change it. So if you had like a frame shift mutation and dramatically changed the amino acids or shortened it or something, by all means you could change it. But if you just change one out because it's not based on R groups, it may not necessarily be changed. So I actually am going to add, just to kind of clarify here, R groups not used. So this is not related to R group bonding. So as a result, changing the amino acid doesn't dramatically change it, but because of the fact that these are weak, they can break in extreme environments, which would alter the shape. Usually when we refer to denaturation, we refer to like the tertiary structure because that's the structure that's so specifically unique to that protein. But these would be prone to denaturing as well because again, they're weak bonds, so the bonds can break in extreme environments. Secondary structure is a lot of like knowledge for not a lot of importance as far as I'm concerned because of the consistency of the pattern. It doesn't tend to be a like really strong determinant of the protein's overall shape. So need to know about it, but more significant then is going to be our tertiary structure. So our tertiary structure is going to be unique folding. So this is consistent in pattern forming. So this is going to be unique folds, so unique bonding and folding of a protein based on the specific R groups that are in that polypeptide. So this is going to be unique folding created by bonds between the specific R groups of that polypeptide. So the key thing that we want to focus on here is that it is unique. So this is going to be what actually gives each protein its unique function, comes from the uniqueness of this folding, and that it is specific to the R groups. So that's kind of the key. I find tertiary structure really hard to draw, so I'm going to just try my best. In class, I did try to draw something that then labeled and showed each of them. So I'm going to actually cheat and come back and draw them purple because I really can't do it when I try to. So this would be like this one here, and then maybe this one would go inwards, up, down, but then this is going to pull back in. This might come out, but then this one pulls back in, and this one might go like that, and then come all the way up here. So there's my little folded thing, which probably in hindsight looks a little bit too much like my Beta pleated sheet. That is not intentional. Maybe I'll make it go a different direction so that it doesn't. So that might go like this. And then finally, like that. Okay. The point is, it shouldn't look like any set pattern. It should look like a jumbled mess. So if it looks like a jumbled mess, it's been a success. Let's talk about the type of bonds though. So it has a lot of different type of bonds involved. So we'll start with the strongest bonds that it has are actually a unique. So I'm gonna just do this from kind of strongest to weakest. So the bonds vary in strength. So it does actually have the ability to form strong covalent bonds. So those are going to be disulfide bridges. And those are those are going to be strong covalent bonds, but they only form between two cysteines. So if you have two cysteine molecules, then they can form a disulfide bridge. So let's say that this was a cysteine and this was a cysteine, it can form that nice strong disulfide bridge. The next thing that can happen is you can form hydrogen bonds. So hydrogen bonds, we'll do ionic next. So the next thing can be ionic bonds. So those are the next strongest. So ionic bonds between two oppositely charged R groups. We're also going from strength to commonality. So although these are really strong, they're actually not super common because there's only 5 charged R groups and it has to be an opposite charge. So they're a little bit hard to come by. But let's say this one is a positively charged and this one is a negatively charged. They can form an ionic bond there between them. Next, we have hydrogen bonds. So hydrogen bonds are going to form, and again, these are weaker. So these are going to be weaker bonds between two polar R groups. and there are 9 total amino acids. Is that right? Six amino acids that are polar. So again, not super common, but more than the others because they doesn't have to be the opposite. So let's say this one, where can I draw another one? I'm going to cheat and extend my chain like that. I don't even know what I'm drawing here. Okay, and then I'm going to draw a hydrogen bond like here. And let's say this has a slight positive and this has a slight negative. So there's a hydrogen bond. And then the third type, which is the weakest of them all, are called hydrophobic interactions. So hydrophobic interactions are not actually a true bond, but it is the tendency for non-polar R groups to turn away from water. So they end up moving towards each other. And usually that is actually towards the center to move away from water. So what they actually are doing is moving both independently moving inwards, so that pulls them towards each other, but the attraction is actually away from water as opposed to each other. So for instance, like this one is moving in to get away from water, and this one is also moving in to move away from water. So they've both pulled in, so that's called a hydrophobic interaction, but there's no actual bond between them. So there are other kind of categories, but those are the 4 you need to know. And you do need to have some sense of their strength. So hydrophobic interactions are fairly weak, again, because there's not an actual bond. And if you put it in a different aqueous environment, like move it from an aqueous environment to a less aqueous, it can completely go away. So those are kind of the weakest of them. So in terms of what impacts them, these are impacted by 2 things. So first of all, mutations, because a change in your amino acid will change the R groups. So if you change the primary structure, then that changes the R groups, and that's going to change your folds. It does depend on the change. So if you replace like one hydrophobic amino acid with another hydrophobic interaction, it may actually not really change the shape. So there's a little bit of variation there, but as a general statement, we will say that it does. And then these are very influenced by denaturation, which we'll talk a little bit about separately, but basically any sort of extremes in temperature tend to break hydrogen bonds. And then changing the pH alter the ionic bonds. So when we alter the pH, a lot of times the addition of new like hydrogen or OH groups will actually mess up the existing ionic bonds, and that will also cause it to change its shape. Okay, so the last one, and I'm going to cheat slightly just because I don't want to erase this whole thing, and I'm going to draw it and I'll just erase part of that. So the last structure is going to be quaternary, and the reason I'm not necessarily worried about that is because literally on my chart I wrote same as above, so the bonds and the impact is the same type of bonds, therefore the same things impact it. So it's very similar on that front, so I'm just going to cheat a little bit and write same and same for those categories because they are the exact same types. Okay, so quaternary, this time by the end of tertiary, we have our nicely folded polypeptide. Some proteins are done at tertiary. So for some proteins, that's it. That's done. But for some, they actually then combine with another polypeptide. So some have this. So some proteins are just one polypeptide, and some are multiple. And then even with like fibrous proteins, they may actually have very little tertiary bonds, and it might be more about the quaternary bonds. So there's a bit of variation in all of this, and that's okay. So this time it is going to be the coming together of multiple, it can just be 2, but of many I'll say polypeptides. So it's usually anywhere from like 2 to 4, so it's not tons, 2 to 4-ish. It's gonna mess up my drawing. And then plus non-protein parts if conjugated. So if it's non-conjugated, it's only polypeptides. If it's a conjugated protein, it's going to have the non-protein parts as well. Really, the whole purpose of knowing that is to know the example of heme groups. So that's kind of the only example you really need to worry about. And that's done by R group bonds between the polypeptides. So it's basically the same concept as here. Instead of the R groups within a protein doing that, it's R groups between the two that are doing that. So this time, here's my, again, I can't color it at the same time, so we'll draw this protein. Okay, so there's protein 1. Ta-da! And then let's draw polypeptide 2 over here. So this one is gonna bond with it. Like so. And so I'll just draw this one with different colors just to show that it is a different chain. So those are my 2 polypeptides, and then some of the same bonds can form. So let's say this was a cysteine and a cysteine. You can actually form a disulfide bridge. Let's say that these are 2, this could be my ionic bond, like here, and then maybe these 2 have a polar bond or a hydrogen bond. So same type of bonds, it's just between the 2 chains. So that's kind of the key. So I'm just gonna put here, same bond types. I don't even know if that's showing up on my video, but it's not. Same bond types as above, and then I'm just going to write here as above because all the same things are true. If you change the R group, you're going to change which R groups can interact with the other polypeptide chain. Okay, so that's a protein structure. Probably the 2 most important ones to really understand for their implications are primary and tertiary. Quaternary is basically just the same as quaternary and tertiary are basically the same, just within versus between. So that's the distinction there. And tertiary, secondary, you just need to memorize the difference between alpha and helix and beta sheet. Okay, last little thing on protein is I just want to talk a little bit more about denaturing and also spell out the connection between structure and function, just because it's really important, particularly as an example of proteins or enzymes, and denaturation is a really important enzyme concept, but we'll talk about it for proteins because it is actually true of any protein. So it's not just enzymes that denature, any protein can denature. All right. And this is the remaining thing we're going to do on this sheet, so use as much space as you have left. If you made your first chart really tiny, you can make this one giant. Okay, so I'm going to label this denaturation. So I'm going to say protein function and denaturation. As kind of a general term for what we're talking about here. Okay, so we are going to start, and so I'm going to kind of go through a little chain, and I did this kind of similar in class, but how all these things are related. So the primary structure of a protein, so in other words, which actual amino acids you have, is going to determine both the tertiary and quaternary structure. And remember, quaternary, it may not have that in every case. A lot of enzymes are actually primarily tertiary only, so that's kind of and if they have it, doesn't always mean they do. And then that is going to determine the shape. So because that structure is all the folds, that is going to determine the unique shape of the protein. And its unique shape is important because it's going to determine what it's compatible with. So most proteins act with another thing. So it might be the receptor, it might be the substrate. In some way, it's going to change something like that. So that's going to impact its compatibility. So that's going to impact its compatibility to a receptor, or to a substrate, or to that part of the membrane. So we'll talk about those as an example. So in some way, its shape makes it compatible for its job. Therefore, by making it compatible to the right thing, that is going to determine its function. So what it is compatible to is what gives it its function. So shape determines function, but we want to go back and say structure determines shape determines function. So a couple things to clarify here. So this is caused by the chemical properties of the R group. So the chemical properties of the R group is what determines how they actually move and interact. And so we'll just do one example because I didn't think we explained it super well. So we'll just say, for example, nonpolar R groups are hydrophobic, and they actually turn inwards. And so they are going to generally move to the center of the protein. And that's going to be important in kind of a couple different places, but this actually plays a big role in how they embed themselves in the cell membrane. So this is really important for integral proteins in the membrane. So integral cell membrane proteins. And that is actually what makes them amphipathic. So what they actually do is they have that, the hydrophobic ones pull in, that gives them a hydrophobic central region that can sit in the cell membrane, and then puts the polar ones on the ends, which allows them to stay with the heads of the membrane. Again, that's going to come up more when we actually get to the cell membrane. So it's not necessarily like super crucial right now, but it's just something to remember as we talk about types of proteins, that those membrane proteins are important. And one of the ways they can be amphipathic is because the clustering together of the nonpolar parts. Okay, so, but because if we talk about these actual quaternary structure, one of the things that we said is that they do consist of a number of weaker bonds. So this is actually made up of some weaker bonds, or some kind of very specific ionic bonds. So because of that, if we actually change the environment, so if we have a dramatic change in the environment, it can impact these. So any sort of dramatic environmental change can damage those weaker bonds. And the two main examples that you would need to be able to account for that can do this is basically a change in temperature. So if you change, that symbol means change. So if you change temperature, you can actually break the hydrogen bonds. And so it will start to cause an unfolding. If you change the pH, it can actually alter the ionic bonds. So it has more of an impact on that. It can also affect the hydrogen bonds as well, just has that particular impact. And the result of that is that it changes the shape of the protein. And that term is to denature. So denature the protein, or denaturation if you're trying to talk about it in kind of noun form, but that is to denature the protein. And if you change the shape, if we kind of follow here, if you change the shape, so if you change the shape, the result is going to also be that it is going to alter its compatibility. And if you alter its compatibility, then the impact is going to be often a loss of function. So essentially, denaturation is impacting the tertiary and quaternary structure by some sort of altering of the temperature or pH, causing a change in shape, losing its compatibility, and therefore loss of function. One thing to note, though, is that it is temporary. So it will regain its shape when you put it back into an environment that is acceptable. So the bonds will reform. So it's a temporary loss of that. Okay, so that's all for B1. In my class, we are testing C1.1 with this. So I'm going to do, next video is going to be C1.1, and that will go with it. But if, in terms of revising just B1, it is just carbs, lipids, and proteins. And sorry, I don't think I did a particularly fabulous job. I'm normally better about this. I just totally forgot to point out that essentially this whole chart is higher level only. So protein structure is a higher level component. So the information on the first video was standard level, so the stuff in terms of amino acids and condensation reaction and so forth. But in a little bit of this, to be honest, is probably standard level as well. But this whole chart is actually higher level. So I didn't mark that, but if you want to make sure to mark higher level here, everything on this first page is standard level. So all SL, but this is actually the higher level component to it. So, all right, thanks so much.